I got a rockin' paper for you. I got a rockin' paper for you. I got a rockin' paper for you. Good evening, Pittsburgh and universe. This is JC on a bike. My name is JC and this is Journal Club on a bike. I got a rockin' paper for you today that ended up in Cell. I think the first time I saw it at BioArchive. And it's from the Sawtell Lab, Nathan Sawtell's lab in Columbia University. And what's cool about this lab is that they use the Mormyrid fish, an electric fish, as their model system. Mormyrids are a real cool aquarium fish that I actually kept when I was a kid. They have an electric organ in their tail, and like an electric eel or any other electric animal in the water, their challenge is to try and ignore the electrical signals that they themselves generate, while at the same time being able to sense the electrical signals from prey animals, which are orders of magnitude smaller than the signal that their body generates. And so this lab showed in a neuron paper earlier this year that somewhere in the electrosensory lobe, the organ that senses the electrical signals for the animal, that in that lobe, which is a cerebellum-like structure, in that lobe, somewhere in that network is generated a negative signal, a sort of negative inverted image of the sensory input that's generated by its own electric organ. And this paper then takes a look at the network and tries to figure out which cells are generating that negative signal and how exactly they're generating it. So I'm not going to be able to get through this paper in an adequate way to do it justice. The paper is extremely strong and very thorough in terms of its dissection of the mechanism. What I want to do is just give you a brief intro that will spark you to look into these two papers in detail. And I wanted to go through them because I did mention them in my previous video with regards to layer 2-3 pyramids and pyramidal cells in general and multi-layer networks. And I think the main sort of take-home message, aside from the observation in this electric fish, is that the apical dendrite, or the long dendrites of cells in neuronal networks, if they are properly compartmentalized electrotonically, the soma and the dendrites, then the typical problem in artificial networks and multi-layer artificial networks is that the network has to be in two different states. It's either in a state of learning so that the synapses can change and become better at whatever they're doing, or they are signaling and processing the result. And you can't really, in artificial networks at least, you can't really simultaneously learn and signal. And it would appear that the electrosensing organ of this electric fish is somehow capable of both sensing and learning, signaling and learning at the same time. And so how this is implemented in a biological system is what's key, right? Because understanding how it works in an artificial network, we've used artificial multi-layer networks to master the game Go and master chess. Wow. And you can't pull the hood off of those networks to find out the secret, right? You just know that you have a network that can beat the best Go players in the world. But we don't know why that network is so good. because the connections and the distributed learning in that network, although we can open up the hood, we can't really interpret anything there. And from a biological perspective then, one of the tricks has always been, so how is a multi-layered network implemented in a biological system? And so in this paper, given this cool observation of the canceling of the sensory input in this lobe, this paper really takes the lobe and dissects the network and finds a plausible mechanism which, when modeled, works very well on how two populations of medium ganglion cells in this model can project onto excitatory or inhibitory output neurons to collectively sort of generate this cancellation of the self-generated signal and allow the emergence of the prey-generated signal. And so in figure 1, they just show you the basic setup. You can see that the muscles that generate the electric signal in the fish are in the tail, but the cartoon shows them trying to sense a small prey animal in front of them. And so the idea is that there's an electric signal from a prey animal which is much smaller than the electric signal that they themselves generate. Somewhere inside the electrosensory lobe, a negative image of their self signal is generated and superimposed on the signal that the sensory organ gets, and the result is a self-signal subtracted prey signal. And so the trick here is to see that there's a corollary discharge from an organ which is, corresponding to this self-generated signal. And that's passed through granule cells to the medium ganglion cells, and then the medium ganglion cells do something to that signal before they pass it on to the output layer. And the medium ganglion cells are all GABAergic, and the output layer is composed of a mixed population of excitatory and inhibitory cells. So the idea is, how do these two populations work together to generate the prey signal that is useful to the animal? So figure 2 dissects the medium ganglion cells can fire broad spikes and narrow spikes. And with careful dissection, they're able to show that the broad spikes are all canceled out, but the narrow spikes from the medium ganglion cells don't appear to be canceled out. So this spurs them to investigate what information might be carried by the narrow spikes. And so in doing so, they, in figure 3, end up working this out to a little bit farther, where they can show that very specifically during learning, the narrow spikes are preserved. And so it would seem that the somatic signals, the narrow spikes, are the signaling, and then the broad spikes that occur in the dendrite are responsible for regulating the plasticity at the granule cell synapse that's at those long dendrites. So figure 4 then takes this one step further and notices that the medium ganglion cells show two different response polarities to a stimulus. One can be excitation and 2 can be inhibition, a decrease in firing or an increase in firing in response to. So that separation allows them, gives them this idea that what if we separate the positive and the negatives, what if the positive sloped cancellation goes to the excitatory cells and the negative ones go to the inhibitory output cells. And so this two-layer network learns specifically for the inhibition and the excitation of the output layers. And this hypothesis is illustrated in figure 4 or figure 3H, I believe. Or figure 4H it is. And so that's their model. That's their model of how this works. Holy cow, thanks for joining me on my ride. That was a real complicated paper by the Sawtell Lab at the University of Columbia in New York. They work in the Mormyrid fish, a spectacularly cool model organism where they show a multi-layer network, a biological example of how a multi-layer network might be implemented. It's a very detailed paper that I won't go too much into now, but suffice it to say that what they've shown is that if you can compartmentalize the soma and the dendrites enough, you can essentially install a hidden layer into dendrites and make it so that what previously wasn't really understood how it would be biologically implemented, a network which can learn and signal simultaneously. This is not something that artificial networks are able to do, and the way that artificial networks overcome this problem is either be in a state of signaling or be in a state of learning, and this is a sort of biologically plausible solution to that problem by compartmentalizing the neuron and putting plasticity in the dendrites while keeping signaling intact in the soma. Again, it's going to take your own work to really dissect this paper and get the most out of it, but what I really wanted to do is just plug it, because I think this paper and the previous one in Neuron are just excellent examples of how we can make progress in understanding how the brain works without having to do a bunch of correlations to 10,000 neurons and to events that the experimenter observes, but rather taking the input and the output, hijacking them both, and then learning how they work inside of the machine. And I think that's what's so cool about this paper, is that they've taken this model system and really exploited it in a way that allows them to probe how it's learning. And this, finding a way to do this in another vertebrate like a mouse will be very, very difficult. And given the fact that, again, this cancellation of self-generated sensory signals is something that in an electrical animal is a very tractable idea, whereas in a more complicated, less obvious sensory sort of inputs coming in, what we generate is not so obvious, and what we generate is not so easy to take over and to manipulate. Whereas in this fish, because of the unique way that the system is organized, they can both use the system as the model, but then also introduce artificial parts of the system so that they can manipulate the way it learns. Yeah, again, it's too complicated for me to explain, but if you liked it, like and subscribe. Thanks for joining me, and I'll see you next time.